I'd like to introduce Aaron Gittler, a colleague of mine here on the Stanford campus. Aaron is an outstanding molecular geneticist. He's a professor of genetics here at Stanford. He works on a number of neurodegenerative diseases, and we're thrilled to have him working on eye diseases with us that include glaucoma and now optic disc drusen. He has an expertise area in both exome, whole genome sequencing, as well as CRISPR-wide screens. And I'll let Aaron.
Okay, thanks so much, Vinu, and thank you to everyone for the invitation to this very interesting symposium. So I'm a researcher at Stanford interested in mechanisms of neurodegenerative diseases, and I'm also interested in human genetics, and as Vinu mentioned, we've been applying exome sequencing and whole genome sequencing to try and discover genetic modifiers and causative genes for various human diseases. And I'm very eager to offer our help and join forces with you and your colleagues on trying to see if we can contribute to finding genes for ODD. So briefly, some disclosures that may or may not be relevant to the material I'll present today. And the learning objectives are to learn the latest human genetics methods for identifying human disease genes with application to eye diseases, and to learn about how modifier genes can affect penetrance of genetic disease. So I'm definitely an outsider, and I look forward to working together with everyone to learn and to see how we can contribute. As we've heard earlier today, it appears that optic disc drusen is an autosomal dominant disease, and understanding the pattern of inheritance of a genetic disease is important because that helps to give clues about what types of mutations we'll be looking for. And this is a classic example of an autosomal dominant pedigree. This happens to be for Huntington's disease, where you could see that the key feature is that you only require a mutation in one copy of the gene. So any individual who has the disease and the mutation has a 50% chance of passing it on to the next generation. On the other hand, there's a autosomal recessive pattern of inheritance in which it requires 2 mutant copies of a gene in order to present with the disease. And this would then, some person, either parent would have to be a carrier, and then there's a 25% chance of the next generation inheriting the disease. So understanding the pattern of inheritance is important because it suggests the types of mutations that could cause the disease, and then also therapeutics. For example, cystic fibrosis, which is autosomal recessive, it's thought that the mutations are loss of function. So when we discover the first genes for ODD, we hope to then use that to learn about pathways and thinking if a gene mutation is loss of function or gain of function. For cystic fibrosis, it's loss of function, suggesting that replacing the gene or giving or compensating for the loss of the gene could be a therapeutic strategy. On the other hand, with Huntington's disease, which is autosomal dominant, it's thought that the mutations are gain of function. So thinking about strategies to inhibiting some toxic mutant gene has actually entered the clinic. So just that framework will be important for today. So once you have these large pedigrees, they can be very powerful, and you can perform linkage analysis where you're asking, what are the parts of the genome that are associated with the disease that are passed down from generation to generation that are linked to the disease and are associated away from individuals in the family who do not have the disease. And if you can identify those, you can then sort of find the neighborhood in which the gene might be, and then you can track it down that way. And we heard earlier today that this disease appears to be autosomal dominant with low penetrance. I'm actually fascinated by penetrance in terms of research because it suggests that two people could have a mutation and one would present with the disease and one might not. And I think that's very interesting scientifically because it suggests that there could be environmental factors or other genetic modifier modifiers that could influence whether or not someone with a particular variant will present with the disease or not. But because the mutations have decreased penetrance, it will be more of a challenge to actually identify the gene, because you could have individuals in a pedigree that do not have the disease but may have the mutation. It may be somewhat tricky. Other genetic approaches to identifying genes associated with a disease are called genome-wide association studies. And this, for example, is in which you have a large number of cases and a large number of controls, and you scan throughout the genome asking for what regions of the genome are different in the cases versus the controls. And you can look at individual base pairs called SNPs, or single nucleotide polymorphisms. And at any given location, you might find that having a G at this position is way more common in cases versus controls. It suggests that having the G at that position is the risk allele, whereas having an A might be protective. Of course, you can have A at that position and still have the disease, or you could have a G at that position and not have the disease. That's why these are called genome-wide association studies. And when you could perform this analysis on millions of SNPs throughout the entire genome, and you get data that looks something like this. This is called a Manhattan plot, because it looks like the skyline of Manhattan, in which you have on the x-axis the chromosomes, and then you have on the y-axis the p-values associated with each SNP in cases versus controls. Doing this type of analysis, because you're doing millions and millions of tests at the same time, the threshold for genome-wide significance is very high, but then you can basically find regions of the genome that might be associated, and that can start your hunt for the disease gene. These types of analyses have been very powerful in trying genes and that are associated with disease, and for many different traits. And importantly, though, you identify from these SNP that might be different between cases and controls, but it doesn't prove that that actual SNP is what causes the disease. It just gives you like the address on the street or the neighborhood where it is, and it helps you to hunt down what the actual disease-causing SNP is. And this is because genes are located on chromosomes that are inherited in blocks together. So you inherit the genotype SNP along with the other one, which is linked. Once you identify a bunch of these, it's important to try to figure out which ones or how they might affect the expression of the gene or the disease. And you might want to think about focusing on genes that are only expressed in the eye. And there have been recent studies to compare genomes with RNA expression analysis. And you can see that different variants and different genes affect expression in different tissue types in a differential way. So in this example, you might have a specific gene in which you can have a polymorphism that increases expression a lot in the eye, but have lower expression in other tissues, whereas other variants might have the same effect in all tissues. So that is something to keep in mind as well when you're looking at variants. But what happens probably in most cases when you only have one case per family? How are you going to find the disease gene? And some of the efforts that my laboratory has been taking for other diseases, spearheaded by an amazing postdoctoral fellow, Julian Kothui, he's been performing whole exome sequencing to try and solve genetic diseases. And exome sequencing involves sequencing only the parts of the genome that are coding for proteins. These are called exons. So the genes are broken into introns and exons. Exons are what are codes for proteins. And to get the biggest bang for your buck, just focusing on these is where the most damaging mutations will likely occur. And to do this, you can use a technique in which you can shear the DNA and have probes, little baits, literal baits, in which you can use a magnet to pull out just the exons and get rid of the rest, and then just sequence the exons looking for coding changes. And you can align these to a reference genome, and you can call variants and say, this person has a variant here, but not here. Just to put up this slide, just to say how this is challenging, if you just look at the bottom of this table, every person that you would exome sequence would have about 20,000 different variants, total variants. So how are you going to find that needle in the haystack? You could say, okay, let's just look for novel variants, ones that have never been found before in any existing database, like so-called private variants. But you'll find 300 of these. So you could say, okay, then let me just look for ones that cause damage, like stop codons, nonsense. Then you'll find 5 of these, or missense ones, you'll find about 200. So it's always a challenge to prove that the gene variants you find will cause the disease. And we could talk about strategies to overcome these hurdles. A couple examples that we've solved in the lab, this is in collaboration with our colleagues in the Department of Neurology. We found a dominant pedigree in which we found using exome sequencing mutations in DNA JB6 as a cause for limb-girdle muscular dystrophy. And then a recessive pedigree, we found mutations in GMPPB as a cause for that disease. Finding the causes for the recessive ones are a little bit more easy than the dominant ones. What's really is beneficial, and I will talk more about this, for finding the causes of dominant diseases is having distantly related relatives, because that allows you, hopefully the mutation will track across generation, and it'll also track away from things that are just inherited from parent to child. We've not solved everything. So in another collaboration with the Department of Neurology, we've been trying to solve this genetic disorder called Alice in Wonderland syndrome, which I'm sure you're familiar with, in which the patients have altered visual perception. And we've exome sequenced lots of these, many of these generations. We found 50 private, rare, or unknown mutations. We haven't solved it yet. And I think it's because we haven't been able to get to these distantly related individuals, in which I think if we could actually get those samples, it would be powerful. So when we're thinking about these collaborations, international collaborations to solve ODD, it would be really powerful to combine forces and look at large pedigrees and distantly related individuals. Another approach could be to look at what's called a de novo mutation. This is something that, these are very rare in an individual, but they occur in the germline of either dad or mom, and then the individual would have the mutation that could cause the disease. So this would be, the way that you identify them is by sequencing trios. We've done a study on these for ALS, the disease I study, but you can imagine setting up a situation where you would have a patient with ODD and both unaffected parents, and try and figure out a de novo mutation that might have happened that caused the disease in the patient, but either of the parents. I also wanted to talk briefly about ideas for genetic modifiers. I'm very fascinated about modifier genes. So there's an example in the literature in human genetics about African Americans in Texas who were basically resistant to high cholesterol. And it turns out that they have loss of function mutations in a gene called PCSK9. And this has revolutionized therapies for cholesterol treatment, because now companies have developed inhibitors of PCSK9 and as a way of lowering cholesterol. So if you can find loss of function mutations in individuals that might make them resistant to a disease, that could suggest a therapeutic target. And so I think this is a great opportunity for our community, because maybe we could find the PCSK9 for eye diseases. So we want to look for elderly individuals who might harbor eye disease variants, but have lived to late life without disease. And then we could sequence the exomes of these to find null mutations or null variants that might be associated with protection. There is an example of this in Alzheimer's disease, in which individuals in Iceland who have a mutation in APP basically have lifetime protection from Alzheimer's disease. And more of these are coming out. This is some recent studies, and again, looking for protective variants. And I think as the ability to sequence genomes is expanding, we'll be able to find more and more of these. Another interesting resource that you should know about is that there are human knockouts, basically many individuals have been sequenced, and they found that they're harboring knockout genes, knockout mutations in genes. And once we identify genes, we can then recontact those individuals and see if we can perform eye exams or other studies to see their phenotypes. So rather than starting with a phenotype, you can start with a genotype and go back and identify phenotypes. The ability to connect medical records to genomes and exomes has been powerful, and I know that the Optic Drusen Center is set up to do this. And then also the UK Biobank is an amazing resource in which there's deep phenotypic data combined with genome data, and you can identify phenotypes connected to different genetic variations. One very recent study in the last few days that came out of Stanford, in which, using the UK Biobank data, they asked for data, genetic variants that might be associated with lower intraocular pressure, and they identified loss of function mutations in a gene called ANGPTL7, which is protective against intraocular pressure. So this suggests a therapeutic target. So it'd be interesting to use a similar approach for ODD. And in the last few seconds, I just want to end with a potential cautionary tale as we're launching into exome sequencing. We hope that the mutation will be in the exons, but it might not be. So this is a study about a new ALS gene. It was linked by association studies to chromosome 9p21. So there was a race to find what the gene was, and every coding region of genes in that area were sequenced. No mutations were found. Every non-coding region was sequenced. No gene was found. And then they were sort of analyzing some region in the area, and they noticed that there was this nucleotide repeat in a non-coding part of the gene, GGGGCC. It was repetitive. So they started designing a strategy to look at the size of this repeat by amplifying across this region. And they found, so they did this in this part of the pedigree here with dad and mom, 9 and 10, and then 3 offspring. And they found that dad had repeat lengths of 5 and 8. Mom was homozygous for a repeat length of 11. The offspring number 16 inherited the repeat of 8 from dad and got the 11 from mom. But then when they came to 17 and 18, both of them got the repeat of 5 from dad, but they didn't get anything from mom. So this was confusing because it violated like Mendelian inheritance, so it wasn't possible. And it turns out that they were tricked by the mom being homozygous at 11. She actually wasn't. She was heterozygous for 11, and then she had a massive repeat that was refractory to PCR amplification. So when they used another technique, they found this massive expansion, nucleotide repeat expansion. And this is actually now the most common cause for ALS. And it's a massive nucleotide repeat expansion in a non-coding region that was resistant to exome sequencing and next generation sequencing. So we should just keep in mind that it could be a nucleotide repeat expansion in a non-coding or coding part of the gene. When we're doing exome sequencing and things like that, we should keep that in mind. I'll stop there. I just want to say that I'm very excited to work and help out with some of our expertise in sequencing and analysis. It's exciting to hear that Dr. Liao and her colleagues have established an IRB, then ready to collect samples. We can send spit kits outside of Stanford. With all of these human genetic studies, it's really international collaborations that are key. So I'm looking forward to working with everyone. The things we've learned along the way, multi-generation families are powerful. Having distantly related relatives like cousins, aunts, and uncles are also super powerful. And the better the phenotyping to true positives and true negatives, the easier it will be for us to identify this disease gene. So thanks, and these are the take-home points, and happy to participate and answer any questions.
Thanks, Aaron. That was a great 15-minute primer of classical and modern up-to-date genetics. I have a couple of questions that'll take us into the panelist discussion. So my first question is, when we have a multi-generational family, we really know what to do. And then if we have these huge populations, we have the tools now to get some amazing information like you described. In ODD, we're kind of stuck in between sometimes, where we have 100 sporadic cases without any clear familial history. So, with a human genetics approach, how do you approach that kind of a population? What's the toolkit you begin to use?
Great question. So I think the exome sequencing could be powerful. I think you'll have lots of variants, but if you have a large number of probands that you sequence, you could do something called a gene burden test, where you can ask if, rather than finding the same mutation that might track in a family, you might find multiple mutations in the same gene, or an increased burden of variants in this gene versus in cases versus controls. And there are statistical frameworks for doing what's called this burden analysis, where you just look for all the variants in genes or in domains of the protein coding parts of the gene and ask if there's more variants in that gene versus the other ones. Now, again, this is like only a starting point, and then it comes with, you'll have to do experiments like some of the ones that Dr. Sun mentioned in model systems like in mouse or zebrafish, where you test variants found in cases versus controls.
Thanks, Aaron. One of the other questions that Wayne Farmer had sent is, could epigenetics explain the low penetrance of hereditary ODD? And how does one tackle epigenetic questions in this context?
Absolutely. So you have variants in your genome, and they will cause or not cause disease based on where and when and how they're expressed. So if you have, and remember, especially for dominant things, you can have some alleles expressed higher or lower depending on regulation. So for sure, understanding the epigenetic modifications and architecture of the genes that are identified will be important. And there are techniques available to profile the epigenome on a large scale basis. So I think we should add that to our toolkit as we're starting these studies. You can also imagine cell non-autonomous interactions with, like, perhaps the immune system may interact, and there might be environmental triggers that given a particular genetic landscape. So I think all of these will be challenges, but also opportunities for learning about the contributors to this disease.
Great. Maybe we can transition to our panel discussion. I know there's a couple questions left over that would certainly fit, or I think somebody else on the panel could answer. Joyce, did you want me to read out these questions?
Yes, given the high interest on the genetic component, I thought we might as well just go right into it. And I have some questions myself too. So go ahead, Vinu.
Okay. I mean, these go back to the previous sessions. If you want to talk about some genetics, maybe we should keep going on that.
So Aaron, I was wondering, in terms of the needs for identifying genes versus epigenetic factors, can you talk a little bit more about that so the attendees for this meeting can figure out how we could synergize to accomplish these goals?
Great. So the genes, I think we just require genome DNA, either from spit or blood samples, and then we can just profile either exome sequencing or whole genome sequencing. I think that's the first place to start. And then I think epigenome, one could look at lymphoblasts that are derived from blood cells, and we could sort of profile how different genetic variations affect expression of RNA. The caveat to that type of approach is that you're require using like the lymphocytes as like a proxy of it, whether it would be expressed in the eye or not. So sometimes those work, but other times you might need to go to the actual cell type that you're interested in. So that might require studies with iPS cells or other approaches.
My understanding of the blood as a source of this material from mitochondrial diseases is that when you look for heteroplasmy and accumulation of mitochondrial mutations, that the circulating blood cells are actually maybe one of the worst cells to study because they're more rapidly dividing and they have fewer of these mutations. Obviously, we can't access the eyes very easily, but another source, as you mentioned, would be fibroblasts. That's definitely better than blood cells. And I was told that renal, so urine samples, so renal epithelium could be even better than blood as a source of looking for at least heteroplasmy in mitochondrial diseases.
Yeah, I think yes, I think those are the concerns. I think studying the pedigrees, though, you might be able to get a sense of if this is sort of mitochondrial or not. But I think we should keep that in mind when we're collecting samples. But I think the first place to start would be these really powerful pedigrees that you and your colleagues from around the world have been collecting, as well as the individually highly phenotyped cases. And I think it's great to hear the colleagues in Copenhagen are launching a similar study. And I think comparing notes and joining